let us study about monohybrid cross. So monohybrid cross में क्या हुआ था that मेंडल ने जो पी प्लांट जो भी पिक किया था उसके अंदर उसने सिर्फ एक कॉन्ट्रास्टिंग फीचर जो है वो पिक किया, that means one opposing feature. So when one pair of contrasting characters was taken to cross two pea plants, and after crossing the generation that was received, that is also known as F1 progeny, उसमें क्या देखा गया कि जितनी भी F1 progeny के जो भी offsprings हैं, the all pods were obtained green in color, and the contrasting feature that he picked up here is color. You can see in the diagram that he picked up one green pod and one yellow pod, and F1 progeny में जो भी offsprings आए, they all showed green color. Further, if the parents are heterozygous, then phenotypically 3 purple flower and 1 white flower was produced. We can also say that genotypically 1 homozygous, 2 heterozygous dominant and 1 homozygous recessive was received. So this is the figure where you can see in this illustration that if the parents are heterozygous, then how the flowers were produced. Now let us study about dihybrid cross. So in dihybrid cross, a breeding experiment was conducted in which 2 pairs of contrasting characters are taken to cross 2 plants. The 2 pairs that are considered here are the color and the shape. So you can see that in the parental part, the color was yellow and green and the shape was round. It was observed that after the experiment being conducted, the offsprings, that is the F1 progeny that was received, was only showing the characters of the one parental part, that is yellow round. So the seeds of this yellow round F1 progeny was allowed to germinate and they were then left to self-pollinate. After self-fertilization, it was observed that the F2 progeny that was received was having a mix-up traits, that means it was showing characters from both the parents. It can be said thus that phenotypic ratio was found to be 9:3:3:1. That means 9 are round yellow, 3 are round green in F2 progeny, 3 are wrinkled yellow and 1 is wrinkled green. But genotypic ratio was found to be 1:2, ratio 1, ratio 2, ratio 4, ratio 2, ratio 1, ratio 2, ratio 1.